Hello, I'm Rob Bies, and I'm faculty at the University at Buffalo. And I'm presenting today on a complementary vignette to the lecture on population pharmacokinetics. These are broken into multiple parts, and this is part one. Part one will focus on exploratory data analysis and the initial elements that are necessary prior to undertaking a formal population pharmacokinetic analysis. So there's some general steps in a population pharmacokinetic modeling project. The first, of course, is to collect the data. So some experiment is designed, or question is arrived at, an experiment is designed, data are generated, and then we collect the data. One needs to understand exactly how the data were generated. There's data formatting involved, depending on what software or what approach you might use for the analysis. There's an exploratory data analysis step, and then model building approaches. A general schematic is shown in this slide with the general steps for the population pharmacokinetic modeling project. And our part one of our vignette really focuses on this top part here, the research question, experiments, data, and visualization data with a particular focus on the nature of the data and the visualization of the data before we get to things such as model context to explore that will be taken up in a subsequent vignette. Again, we're going to use actually a case study with quetiapine, and these are quetiapine concentration measurements that were used for population pharmacokinetic analysis for data that were generated from the clinical antipsychotic trials of intervention effectiveness. This is an NIMH-funded study that completed over a decade ago. We're going to, again, familiarize ourselves with the study design and understand the data generating process. We're going to go through a very basic illustration of how to construct a data set for a population pharmacokinetic analysis, in this case, a non-mem-ready data set. Non-mem is one of the many software available to conduct this type of analysis. And then we will go through an exploratory data analysis evaluating the different characteristics of the data that were collected. This is done prior to the formal model building process and is often done with a data wizard or with a statistical programming language such as R. Things that we will be evaluating are the numbers of observations, numbers of individuals, in this case, concentration observations, number of individuals, what were the doses administered, what are the patient-specific characteristics, what is the distribution of times after dose for the concentration measurements, and then any information that one can glean from a typical population pharmacokinetic study with respect to the concentration versus time profiles, both at the individual and aggregate levels. This is important in terms of evaluating those profiles at the level of patient-specific or study-specific characteristics. Another part two of the vignette, will cover the population pharmacokinetic model building and analysis strategy, and is outside the scope of this initial vignette. So a key step is the familiarization with the data generating process or the study design. When considering the study design, one should evaluate whether or not this was an observational or a randomized study. This will impact the consideration of potential confounders. Was the study conducted in a highly controlled setting like a clinical research center? What was the concentration sampling paradigm? Was it random? Was it optimized? What was the dosage paradigm? What was the frequency of doses? What were the range of doses? What covariates were collected, i.e. what patient-specific characteristics or demographics do we have in the data set? And then what was the nature of the sample collection? Were biomarkers collected in addition to concentration? was the timing recorded appropriately? So let's move on to the construction of the data set. So when one is assembling data from a study such as Katie's study, one may get data in multiple table formats. Here I have a demographics table showing weight and sex by study subject. I have a table of doses, so you have the study subject. You see that there are multiple doses listed and the time of those doses in hours. And then I have a table of observations. So here we have the study subject, the times, the plasma concentration measured. So you notice the study subject is the linking variable here. And for a population analysis, the study subject, that is the basic level of analysis. One considers that as sort of the basic unit of analysis with respect to the determination of parameters. And so if we look at a typical non-mem data set, it is actually assembled from those components. You see the study subject ID appears at the far left, and different elements here, but we will go through these individually in a moment. Now, sort of superimposed the original table formats, or at least excerpts or subsets of those initial table formats to illustrate this. See that our table that indicates study subject and the dosage histories are captured in the overall analysis data set as the study subject ID, here labeled pound ID, and you can see these are listed here. The dose in milligrams in this case appears in the AMT or amount column, and you can see that they are non-contiguous rows for the most part because there are observations now in between. And so this is now sequenced by time. Similarly, the next table showing study subject, time, and plasma concentration appear again in the ID column. Time and the DV, which in the non-mem parlance stands for dependent variables. This is your observation. This is your concentration. And you can see now that we've got plasma concentrations interspersed here. So the non-mem data set runs sequentially. So you have a dose and you have an observation, and it runs sequentially in time. Time must be ascending. The other thing to note is that we don't have any less than symbols, and we don't have the negative timing. Typically, you do not want to avoid putting in a negative time, although the newer versions of non-mem can manage that. And only numerical elements can be present in the data. So you cannot have a less than symbol. You cannot have alpha characters in the data set. That's mainly because the software uses Fortran, but even the other analytical software for the numerical components will require numerical inputs. Finally, we have the demographic table. So again, study subjects, this is indexing here. Weight, you see weight appears and weight is numerical. And then we have sex. Sex is listed in the original table as female and male, but has to be converted to a numerical flag in order for this to be incorporated into the analysis. And so you see we have a column we're calling sex M, and 0 is female, 1 is male. And you can see these are the first two individuals are listed as female, and these values are noted on every single row for those individuals. So now that we've put together a general data set, there are a couple of other components here that were incorporated. These are the missing dependent variable and the event ID. And these are non-mem specific indicators and sort of beyond the scope of the presentation today. But I will just make a quick note that at least the missing dependent variable is used to indicate when there's an input process or when there's not an observation. So here, when you give a dose, you have a missing dependent variable because you cannot have a dose and an observation, an input and an output in the same row. And so you'll notice the missing dependent variable is 0 when you have a plasma concentration. This is illustrated here. So let's move on to the exploratory data analysis component. We thought about the study design. We formatted the data set, so we're ready to start potentially doing some analyses. And now we're going to do an exploratory data analysis, evaluating the information we have in this data set prior to modeling. We're going to consider the available observations and demographics, the covariate distributions, the correlations, the signal ranges. How broad are those signal ranges? Are they something we want to test because we have enough variety, enough heterogeneity in those measures to be able to pick up a difference if there is one? We're going to evaluate the concentration measurements, the units, the range of observed concentrations, any correlations with covariates, doses administered, the routes of administration and the administration rates, etc. This is also the opportunity to evaluate extreme values in the observations when one does exploratory data analysis, in particular visualizations. These become apparent. One will also want to evaluate the proportion of measurements that fall below the limit of quantitation, the determination of samples that are missing or damaged. So these are two separate components. Below the limit of quantitation means a measurement was made, but the limit of the quantitation of the assay was crossed, and therefore the measurement value may be reported just as below the limit of quantitation, or a number that does not conform to the stringency of the assay as it was developed for quantifying those values. Again, a preliminary evaluation of observations with an evaluation of outliers. Potentially evaluating the concentration time profile if we have sufficient sampling. A preliminary determination of the potential of the structural models to test. And the preliminary calculation of potential parameter values, overall clearance, for example, the volume of distribution, to use as starting values. All population PK analyses are nonlinear regression problems, and you have to start with initial values, initial estimates. So for this exploratory data analysis, as I mentioned, we have the quetiapine concentration measurements in the Controlled Antipsychotics Trial of Intervention Effectiveness, CATIE. This was a treatment study with patients in a schizophrenia arm and an Alzheimer's disease arm, and sparse concentration measurements were taken. We had approximately just over 2 concentration samples per individual. So as we explore the data, some tabular output in terms of the population description. So, number of observations. In the schizophrenia arm, we had 811 concentration observations, and in the Alzheimer's arm, 132, so 943 observations all told. The bottom table shows a distribution by self-reported race, so you can evaluate the relative contributions here and whether or not they appear to be representative of the population at large. One thing to keep in mind as we go through these different elements is whether or not there appear to be systematic differences between the arms of the study, or even amongst the covariates. This may potentially confound identifying a specific effect if there are strong correlations. Similarly, here we have a table at the top showing the distribution by sex in the schizophrenia and the Alzheimer's arms. We had 404 individuals, and we've got 325 in the schizophrenia arm, 79 in the Alzheimer's arm. In the schizophrenia arm, it was predominantly male, almost 76% of the individual patients. In the Alzheimer's study, it was more evenly split, with actually a slight majority of female patients. In terms of smokers and non-smokers, see that we had a majority of smokers in the schizophrenia arm versus the Alzheimer's arm. So this could be an issue because if there's an effect of smoking, you might have smoking and arm confounded because of the different distributions. So this is something to keep in mind when you're exploring explanatory variables. In terms of the weight distribution, you can see as well the schizophrenia arm, the weights were substantially higher. Similarly, for the age distribution in the bottom table, we have Alzheimer's arm, the age was substantially higher, not surprising with respect to the type of study. So again, if you're interested in if you detect an age effect, one has to consider that there may be a different disease process as well. These are different arms of the study, dosages were different. These are all elements to keep in mind. So while tabular approaches are helpful, it also is important to visualize the information. So this next slide, which was created using R and a package called gGalley, shows a matrix plot for patient demographics. This is for both the Alzheimer's and schizophrenia studies combined. So what you see on the diagonal of this matrix are the distribution, for example, for age. So you can see it looks quite bimodal. The distribution for weight, it's a bit of a shoulder here, so maybe not entirely Gaussian. You see also the distribution by sex, female and male. And you see the distribution of smokers in the combined studies. You see that's almost 50-50 if you combine the studies. In the off-diagonal elements, we're able to evaluate correlations and other aspects of the distribution. So you see a relationship with sex and age across the studies, smoking status, and age as well. And similarly, a weight distribution difference for sex, with males being slightly heavier. Smokers and weight do not show, at least for the combined set, this element. In terms of the other off-diagonal elements here, the size of these squares represents the number of individuals. Here we have non-smokers and smokers by sex, so males and females. Okay. And then in the bottom left set of off-diagonal elements, we actually see the individual histograms. So you can see the distribution of values, for example. So for age, by sex. Okay, for weight, right, by sex. Okay. You can see the shift here. These are the males, these are the females. Males typically being a bit heavier, but you can get a sense of signal ranges. Now, one of the major components is that we have these subcategories. So what happens when we subset these characteristics, let's say by sex and smoking? That's what's shown in this next set of plots. And again, on the diagonal, we have the distributions, but this is now by different sex groups. So you can see that we have more females in the older group, but in the younger group, females and males are pretty much matched. Similarly for weight, the males are somewhat heavier. Okay. And then by smoking status, you see that males tend to be more likely to be smokers. Similarly, if we subset by smoking, by smoking and non-smoking, you can see that most of the smokers are in the younger age group. Weights overlap, but we again see this big difference by sex. So many more male smokers, for example. Similarly, you again have the distributions for the individuals, and you can look at it in terms of correlatives. There is a significant difference here. So if we consider sex and age of non-smokers and smokers, there's a similar trend here. So we have typically younger individuals smoking in this study set. By weight, again, weight, and you see that for smokers and non-smokers. In females, the smokers tend to be a little bit lighter than in males, but it doesn't appear to be much difference in weight. So we start to get some insights about the distribution of these parameters. So what if we consider the study arms separately? So actually going back to the previous slide, you can see that there were some, there were some differences. There was some sex and weight differences in sex and smoking status and that sort of thing. So if we go and look at only the schizophrenia arm, notice now that the age distribution is quite different. It's less, it still has a shoulder, but it's less sort of bimodal. And the sex and age differences are less pronounced. Smoking and age differences are less pronounced. Individuals tended to be younger. We still have slightly heavier males and females in this study. And so that might be important information. Is there an effect of the disease, or is it the effect of the age group, or is it effect of the weight if we're looking at specific effects on pharmacokinetic parameters at the population level? We subset this by sex groups or by smoking and non-smoking groups. See that many of these differences are not preserved. That's the previous graph. So again, age, weight, smoking, and then along this matrix here, you see these distributions are largely overlapping, at least in terms of the continuous variables, when you subset along the status within the study. Similarly, you can see some weight difference subset by smoking and non-smoking groups. Non-smokers, smokers, males actually tend to be a bit lighter. What about the Alzheimer's arm? So here again, you see that the age now has shifted to the right. The weight now, again, does not have as much of a shoulder. It's more Gaussian. The sex distribution is more even. And the smoking distribution is quite dramatically different. Very few smokers actually in this study. And if we subset by the sex and smoking status, you see that for the different sex groups, you can see that there's almost no smokers here for female versus male. And that we do have a shift in the weights. Females are lighter than males in this case. Ages, though, are quite closely overlapping. So things to keep in mind in terms of potential confounders. Again, this is done with G-Galley, and it's I think an important evaluation of the data before we jump into the analysis. So those are patient demographics. What about elements that are related to the pharmacokinetics directly? So these tables show doses and the frequency by trial arm. So the top table focuses on the dosage interval. So there were 12-hour dose intervals and 24-hour dose intervals. You can see that in the schizophrenia study, the majority of individuals receive the doses twice a day. 86% of those individuals. 15% receive the doses every 24 hours. In the Alzheimer's arm, no patients receive doses every 12 hours. 100% of patients receive them every 24 hours. Now, this might be of concern if the drug has a relatively fast half-life. Perhaps you'll have more measurements below limit of quantitation because of the dosage interval being wider before the patients are redosed. Overall, if we combine the studies, about 70% of the patients were receiving quetiapine every 12 hours and 32% were receiving them every 24 hours. Some patients had both dose intervals implemented. So they had some periods when they were on every 24 hours and some periods when they were on every 12 hours. What about the doses administered? So this bar chart is showing doses, daily doses for the schizophrenia arm. And you'll see that we have doses that go from 200 to 800 milligrams in the schizophrenia arm. Not very many low doses. So this would be 100 milligrams twice a day, for example. These were all given every 12 hours. And a relatively even distribution across the dosage ranges. So well represented for the analysis. With respect to the Alzheimer's disease arm, you see the dose distribution is quite different. It's all given every 24 hours, and the dosages range from 25 to 200 milligrams daily. And doses are listed here in micrograms, just that matches our units in the actual analysis. And the majority of doses here at the low end, 25 milligrams. So are we going to see many more, again, limit of quantitation issues with the Alzheimer's disease arm? Because they're lower doses over a wider interval. Then again, they're older patients, and maybe they eliminate the doses more slowly. So these are all things to keep in mind with respect to the population pharmacokinetic analysis. Another key element to evaluate is how much information do we have? How many concentration measurements do we have across the entire concentration time profile? We have absorption processes, distribution processes, and elimination processes. And these are sparse samples. So it's important to understand whether or not you have sufficient sampling across the entire dosage interval so that you can inform those parameters. So on the left, this is from the schizophrenia arm. We have the time since last dose distribution with a 12-hour dose interval. We have the frequency on the y-axis and the time since last dose on the x-axis. What you see is that we have quite a few samples shortly after the dose. So we'll get the absorption processes reasonably well, but as we get closer to the end of the interval, we have fewer samples and less information. With respect to the time since last dose distribution for the 24-hour dosage interval, you can see we again have less information actually in the absorption profile, and then we have more information in the tail phase of the distribution with respect to concentration and time profiles. For the Alzheimer's disease study, we see a similarly bimodal distribution, which probably relates to clinics being closed in the middle of the night and patients coming in for usual clinic hours, and depending on whether or not they take their dose in the morning or the evening. Again, time since last dose, we have the frequency on the y-axis, time since last dose on the x-axis, and you see that we have information that will help understand the absorption process, absorption profile, and then the tail distribution as we go towards the end of the dosage interval are reasonably well captured. So what about actual concentration measurements? Well, here's a plot with the dose normalized concentration. You remember we had multiple concentrations. This is for the schizophrenia arm. And each of these black lines represents an individual with the time since last dose on the x-axis here, time since last dose, and they're connected. So you can see profiles for every individual. The green line in the shaded area is basically a smooth through these profiles. So you can see that overall, we capture the upslope and downslope of the concentration time profile that one would expect with this drug, and we can evaluate whether or not there appears to be multi-exponential character. We can also evaluate whether or not this may be sufficient to actually allow us to calculate some preliminary initial estimates for clearance and volume of distribution, for example. Now, there were multiple doses given, though, and maybe there's a nonlinearity in dose. And to evaluate that, again, we have dose normalized concentration on the y-axis and time since last dose on the x-axis. But now we have a color coding for the different doses. Again, for within individuals, we have connectors connecting the doses, and blue, the light blue are the highest doses, the black are the lowest doses, and you can see that these pretty much overlap. So if you normalize the concentrations, it suggests that linear processes are driving the pharmacokinetic profile. Another evaluation is to look at the individual patient's time concentration profile. So this matrix of plots is showing concentration on the y-axis, time since last dose on the x-axis. Again, this is a schizophrenia study for the 12-hour interval. And this is useful for identifying whether or not there appear to be any significant outliers by individual. And we don't really see any here. Similarly, we look at the 24-hour interval. You see, this is for the schizophrenia arm. And again, don't necessarily see any outliers. There are a few, maybe higher concentrations here that we want to investigate. But generally speaking, they look reasonable. For the Alzheimer's disease arm, here's a sample of patients. Again, this is a matrix plot. Each panel represents a different individual. Y-axis is the dose normalized concentration, and the x-axis is the time since last dose. And again, you can use this to inspect for potential outliers. So in conclusion, a complete exploratory data analysis is critical prior to beginning the actual model building process for a population pharmacokinetic analysis. Insights on what types of model structures can be supported, what covariates may be considered, and what potential confounds can be pre-identified. And so now that we've done the exploration of the data, we can begin the population pharmacokinetic analysis. And this will be covered in the vignette part 2. I'd also like to acknowledge 2 individuals who contributed to this. Kristen Bigos is faculty at Johns Hopkins University. She facilitated access and provided some additional analysis on the quetiapine data set, and an MS candidate, master's student at the University of Buffalo, Haran Gao, who prepared many of the graphics that I showed using those R packages. Thank you very much.